Hey all, Miss O'Leary here. I had a few requests to post a mini lecture for folks to review what we talked about in class related to bioenergetics. So I will try to go over the biggest concepts here. Know that depending on what teacher you have, you may have gone into different detail in different sections, but hopefully you will recognize some of the major ideas from all of your classes. Okay, so the main topics that are going to be covered, this should also be a good starting point if you want to review what you know from class. Differentiating spontaneous versus non-spontaneous reactions, looking at ATP and its structure and function, looking at enzymatic coupling, which is one of the jobs that enzymes have in our cells, and then also examining electron carriers. So you've all had some sort of chemistry background, so these should look a little bit familiar to you in terms of the types of reactions that you can have. So the top one, we see some things coming together. The second one, we see things breaking apart. And then the bottom two, we see some swapping of parts. If you consider these molecules, they're swapping with each other, going from one to the other, or even trading. So these reactions are very consistent in what we see in biology, and in particular in the process of metabolism. So when we're talking about energy, we're looking at the capacity to cause change. So that's sort of a nebulous description of it. You probably learned more specific ones, for example, in physics. But in this case, when we talk about work in a cell, we're looking at chemical, so things that are of a chemical nature, making change, transport, which is what it sounds like, and mechanical, which is a little bit more like building and structure and things like that. So one of the major things that we occasionally talk about with metabolism is free energy. So this is the energy that is available to do work. So there are equations and a lot more detail that you can get into in terms of calculations regarding free energy, but we do this all to see whether or not a reaction can happen spontaneously. So spontaneous means kind of what you think it means in social discussion. So basically it doesn't need anything else. It doesn't need any extra input to happen. Basically you can see them as corresponding to one another. So if you have more free energy, there is more capacity for work to be done. A different aspect of that would be equilibrium. So rather than having more of one types of energy, equilibrium is talking about something where there's maximum stability. This is where you would have the least amount of free energy. So systems at equilibrium cannot actually do work, which we'll look at a little more in the next few slides. And you can do some changing of our system, though, if you look at a potential input of free energy. So these pictures should help a little bit in what we're talking about related to energy. So this is a hydroelectric system. So you can see that if there's more water in one tank, it will flow down towards the other, and it'll turn this turbine. So this is where the work is happening here. Delta G, this is, if you remember, I referred to calculations and stuff like that. This is what we're talking about with free energy. And so there's basically a flow this way. Now, if you get to a place where these two containers are equal, the turbine no longer goes. So this is an example where you see that if they're in equilibrium, no work can happen. So nothing's happening because there's not movement of water from one to the other. But even in this first one, eventually you're going to get from point A to point B here. So these will eventually even out. But if you have a continual input on one end, so for example, the faucet here, the faucet will continually add water to one side so that work can continue to happen as the water flows to the other one. So if you think about biological organisms, so like our little bunny down here, if you were at equilibrium, no work would be done. And if you can't do any work in your cells, so there's not an excess input of energy, then that means death. So equilibrium biology, not great. But if you have a continual input, so for example, our faucet here has to do with our input of nutrients. So think of the faucet like eating. So if you keep putting food in, then you keep having the ability to do the work that your cells need. Cellular pathways tend to do this in a multi-step process. So instead of just going from the faucet all the way down to the base here, there's usually multiple places where work can happen. Breaking it up can be a little bit easier overall. And then also another benefit is that regulation can happen at each step of this system. So we have these cute pictures here to help us remember these different types of metabolic processes that we will talk about in terms of doing work. The first is catabolic, so think cat. Cat breaks down the fish. So catabolic, we're breaking down a molecule. So think about when you eat a piece of bread, for example, the bread itself doesn't go, through your blood vessels into your muscles. The bread gets broken down into its smallest parts that can be used to convert to available energy for your cells. On the opposite side here, we have this person that is knitting something. So if you think about it this way, every time you knit a little piece, you're doing something. So you're taking a step or having a reaction happen. And a series of them will eventually make this bigger product. So you can't go from your knitting needles doing one step to a whole scarf, for example. You're building it by pieces. And we do see both of these happen in metabolism. So metabolism includes breaking down things to make usable energy, and then also using energy to build up what your cells need. Okay, so to shift gears a little bit to talk about how does this contribute to our understanding of spontaneous reactions. So I live by the Kishwaukee River, so my silly example has always been that if I'm walking with my husband by the Kishwaukee, I can spontaneously decide to go swimming. Okay, I can spontaneously decide to go swimming. I don't need anything extra. I could just jump in the water like this person in this picture here. I couldn't spontaneously decide to go kayaking because I would need something extra. So if that helps kind of remember spontaneous versus non-spontaneous. And spontaneous also favors the products. So if you're on a diving board, more specifically, that's going to favor jumping off the diving board. Let's say that these little molecules are floating in a liquid, they will naturally spread apart spontaneously if there's nothing that's keeping them together. Some chemical structures will break apart spontaneously. When we get to enzyme coupling, this will be important also, but if you go back to my example of my husband and I walking by the Kishwaukee River, you could think of maybe my husband being the enzyme. If he gives me a little push and pushes me in the river, that's causing something spontaneous to happen at a specific time in a specific place. And yes, I would yell at him, but it's a good example of how you can have something spontaneous that is still facilitated. Okay, so next we're going to shift gears a little bit and talk about a very specific chemical structure that relates to spontaneous reactions, ATP. So ATP is broken down into ADP and phosphate. So ATP is adenosine triphosphate, and that refers to the three phosphates here. The adenosine looks very much like what you'll see in a DNA structure. So if you would circle all this part over here, you have the sugar, there's a nitrogenous base here and a phosphate. So this looks like an A in the ATCGs of DNA, which we'll learn about later, but it has three phosphates on it. So if you turn it into ADP, that's a diphosphate. So that would mean that you cut off one of these phosphates. So that gives you the PO4 3-, and then you'd have your ADP over here. Now, if you look at these, you'll notice that these phosphate groups all have negatives on them. And we know kind of naturally that opposite charges attract, but like charges repel. So if you think of it, you have these things that are repelling each other right next to each other in this structure. So this structure is already under some tension. So that means it's going to be pretty easy to pop one of these phosphates off of it. And that's why we would consider this reaction that's written up here as a spontaneous reaction. Doesn't need any extra input. There's already basically flexing here because of the negatives next to each other. On the other hand, if you had a phosphate and an ADP and you wanted to send this arrow the other way and make ATP, well, now you're having to put those negatives next to each other. Think about that taking more work. It's going to take more effort. And so that's non-spontaneous. So the reverse of that is a non-spontaneous reaction. This is helpful because if you think of where available energy is, a lot of it has to do with electrons and moving bonds and things like that. So having ATP as a ready source of a spontaneous reaction will help us with some of the next concepts that we talk about. And the next concept is enzymatic coupling. So you've probably talked a little bit about enzymes being catalysts, so they speed up a reaction. The general definition is always that it lowers activation energy. When I push a little further, people can't really always tell me what activation energy is, but it's really just making the environment appropriate. So if you think about the ATP being kind of like a stick, there's already some flexing in it, it's already flexible. It would be very easy to kind of put some pressure on it and break that phosphate off. So that breaking of the phosphate could be catalyzed by an enzyme. Okay, so an enzyme could do that. And so an enzyme, if you're more of a visual person, you can see that this is like the activation energy that you would need normally, like to break down a particular substance. But if you have an enzyme and it's catalyzed, it drops how much energy is actually needed. So what does this have to do with spontaneous reactions? Enzymes can really only facilitate spontaneous reactions directly. But if you think of spontaneous reactions happening on their own, and they can release energy, for example, with the ATP that's stored in these bonds here, if you think of the bonds storing energy, if that's released, those things can both be used to actually power non-spontaneous reactions. And so what an enzyme does is it puts the things for a spontaneous reaction with the things needed for the non-spontaneous reaction. This is a little confusing, but if we look at some examples, this should help out a little bit. So spontaneous reactions, again, tend to favor the products. So, like the diving board, it favored the person jumping into the water. So if we think of this as being a reaction going from A to B, it's favoring B. The non-spontaneous reaction favors the reactants. So if we're going C to D, it's favoring C. So separately, they have these ratios to them. If you put them together, however, you can see that we've decreased B here and increased D. And so basically we're getting more D out at the end than we would over here. So if our goal is to produce D, we're going to be able to get more of it if we put it together with the A to B transition. Okay. And this is, you can read it down here. So the quantities of B and D in this case are equal because as you create one, you also create the other, as opposed to this in the separate reactions. So to look at a specific example that will kind of tie all this together, ATP breaking down to ADP and phosphate, that is spontaneous. So remember, because of those phosphates, so we got a lot of this as a product. On the other hand, putting two amino acids together, so hopefully you remember this is dehydration synthesis, this makes the primary structure. So linking those together is a non-spontaneous reaction. However, what you can do is put the breakdown of ATP together with the linking of the amino acids. And so you get this dipeptide product out from the end. In this case, if you remember your enzymatic coupling for this specific reaction, this is dehydration synthesis and it takes place in the ribosome. So in this case, the ribosome is the enzyme and it's bringing ATP and amino acids together so that both of these reactions can happen at the same time. And that facilitates this non-spontaneous reaction because it's allowing this reaction to happen right next to it. Okay, so to see another one that is another example, this one is specific to one of the first steps in glycolysis. So again, we said favoring the products here, so ATP breaking down. And then the first step in glycolysis is having a glucose and a phosphate come together to make a glucose 6-phosphate. So that's basically a glucose with a phosphate group stuck to it. Well, the first reaction here will give you a release of energy because you're stored in the bonds because you're breaking down the ATP and a phosphate. And if you look down here, you need one of those phosphates and we need to basically push this arrow to the right so that we can actually get how much glucose 6-phosphate we need. So when you couple them together, the reaction looks like this. You put the ATP and the glucose together, and then the ATP will break down on its own, and as it's doing that, it will transfer the phosphate to the glucose. So this phosphate, you can see over here up on the top, you get a phosphate that will attach to the glucose. So it'll break off the ATP and basically switch the bonds. So instead of being attached to the ADP, the phosphate will now be attached to the glucose. And so there's a lot of these examples of enzymatic coupling in metabolism. So a process that's spontaneous that drives a process that is non-spontaneous. Okay, and the final type of bioenergetics information we're going to look at is redox reactions. So redox is short for reduction and oxidation. And this is looking specifically at the transfer of electrons between molecules. And so the easy way to remember this is Leo says GER, or oil rig. Oil rig is the one that helps me the most. It is a little bit backwards from what you might think. Oxidation is loss, reduction is gain. So think of reduction of available electrons on the outside. So you're putting it into something. So if you look down here in our example with lactate, lactate is oxidized. So that means that you can break off electrons from it. So you're taking electrons out of it. And at the same time, that electron can be passed along to the NAD+, which is reduced. So you have an electron that started on one side, and now it's one molecule is oxidized, and then the one that the electron moves to is reduced. And in particular here, we have NAD+. You do have to also remember from your chemistry days that you have to balance out your electrons and protons. So that's why we see an NAD+ becoming an NADH here. And we talk about redox reactions in particular because this is what helps us understand the function of electron carriers. So an electron carrier is kind of what it sounds like. It's a little bit like if you have an Amazon truck. If you order a lot of stuff off of Amazon, like I do right now because I don't want to go to the store by all the germy people, I'll have Amazon, I'll drop off a package for me, and then it goes back to the warehouse and it picks up another one, and then it comes over and it drops it off. So an electron carrier is kind of like that. So it can pick up electrons from one place, and then wherever it goes, it can drop off the electrons again. So in this case, we're looking at NAD+, so that's what this picture on the left is. If you reduce NAD+, you're going to add an electron to it, which will be balanced out, okay, by your proton. So 2 electrons, and you'll get a proton. You can see those down here. So NAD+, when you reduce it, becomes NADH. So now this is my Amazon truck with the package in it. When the Amazon truck gets to my house, or the NADH gets to where it's going, it gets oxidized, and so then it will drop off these electrons, and because it drops off the electrons, it loses its proton, and so now it's turned back into NAD+. So there's a couple different electron carriers in metabolism. NADH is one of them. You will see a few other ones in photosynthesis and respiration. But the important thing is that they're carrying electrons, and they do this through reduction, picking up the electron, and oxidation, dropping off the electron. Okay, there is kind of 2 halves to this reaction. This is a little bit of pretty much what I just said here, but you can split up this reaction into 2 parts. So first of all, the reduction is half of it, so the NAD+ taking the electron, and then the other half is the oxidation, where something is losing the electron. So often you'll see half reactions referred to when you look at redox reactions. One of them is the reduction part, one of them is the oxidation. Okay, so that was a lot of content, but just to review, we're using all of this bioenergetics to understand metabolism, which is really all of the chemical reactions that happen in an organism. We looked at the difference between spontaneous and non-spontaneous reactions. We looked at ATP specifically and why its breakdown is spontaneous, and how that can help us through things like enzymatic coupling. And then we also looked at electron carriers and how they can function in these processes by moving electrons from one place to another. So hopefully that helps a little bit. Please bring questions to your classes so that you can ask more specific things if you have them. And then hopefully your application as we go through photosynthesis and respiration will make these concepts even more clear with some specific examples.